also important in defense and injury, in tissue repair. The issue of clotting factors. The blood also has what we are calling clotting factors. One that makes blood clot. So that at least the one that's when the thrombocytes or the platelets makes a mesh, but you need the clotting factor so that when the cells are trying to move, they are caught in there. The clotting factor able to clot so that to seal, that you form a prevents damage of it. So the three functions that it does. But our interest is to look at the. Remember, our interest is in how is blood made. That is our interest. Now it has a liquid portion that is plasma. What we are, you have the leukocytes and the thrombocytes. So you have that is plasma, and then you have the cellular component where you have the red blood cells, platelets. Now it's important to talk about when you are talking about whole blood. Whole blood here. I mean, what you be learning, we'll be talking about whole blood. Whole blood is it's the blood that is circulating in the system, or the blood that you've collected in a tube that contains plasma. It contains the cells and other constituents. That is whole blood. Now, when that is whole blood, but again, there's what we are calling peripheral blood. This is blood that actually you are able to collect from the veins that we're talking about. So the blood that we collect actually, that is peripheral blood. The blood that we are able to collect and put in the veins, the one you can access through the veins, that is peripheral blood. So whole blood, that is the composition. Now, I don't know if the simple diagram that I've put there is very clear. So percentage by volume, as we shall see when we are talking about plasma. There in the structure, you are saying plasma is 55%. Then these others, the blood and other constituents is almost 45%. Now, plasma, you have 10% proteins, you have 91% water. Now, what constitutes these proteins? You have what you are calling the albumins, the glutamins, and the fibrinogen. So these proteins are very important for transportation of things. Transportation of albumin is the one that transports electrolyte into bound. Oxygen to be. Of other constituents at about 2%. Now, these other constituents, nutrients, products, the cells, and the regulatory substances, the clotting factors that we're talking about. Now, when you come to blood, you find that actually blood has a component of platelets. It's about 200 to 500,000. Leukocytes, you have about 6 to 17,000. Then you have blood itself in those levels. Then from there, now leukocytes, if they are 6 to 17,000, it's a range. We are saying the volume of blood. One thing that I forgot to mention is the bigger the animal, the bigger the volume, because if you are saying 77% of a cat that is weighing 3 kgs and 7% of an elephant that is weighing almost 4 tons or 3 tons, it differs with the bigger the animal, the bigger the volume. Now, because of that bigger volume, you expect that even these percentages of plasma, it will fluctuate between 55, 45, and somewhere there, it will be a range. Why is it a range? Because it differs from species to species. Even within the same species, we find that actually, even for us human beings, males and female, male versus female, we find that males have less volume compared to females. That is what we are. So that is the composition of blood. Plasma, cellular component. But maybe we can try to add a bit of flesh to this stuff. Now, as I was saying, we are saying plasma, that is the range, depending on the species of the animal, the size of its red blood cells. So you find that in some animals, animals have bigger red blood.
Is it my phone?
Sir.
Yeah, sorry, my network. Yes.
Yeah, you repeat what you are saying. You are not getting it.
Yeah, I saw it. My network was disconnected. So I was saying, plasma, they are those from 5 to 78%. That is just a range, depending on the volume, the sample volume, and also depending on the species of the animal, and also the size of its red blood cells. So I was saying, within these are our animals, they differ. You find that a dog will have more bigger red blood cells as compared to a pig and a goat. So definitely the volume, even if they can be of the same size, but the volumes will differ based on the size of the red blood cells. And also the species. You cannot compare the volume, because even within the same species, let's take for instance bovine. From beef production ideology of the smallest breed in Zambia is the Amboni, not so. So even if it's cattle, even if we are to get the same breed, the biggest breed is the Barossa. Even if you get the two of the same age, same size, same age, you find that they will differ in sizes. Definitely within the same species, also the volume will differ. So we are saying 99% of plasma is water, and then the substance dissolved or suspended in the plasma. Those are the elements about the globulins and the fibrinogen. Then you have oxygen, carbon dioxide, nitrogens, you have lipids, you have amino acids, metabolic waste, and electrolytes. Electrolytes here, you're talking about the sodium, the potassium, the chlorine, the magnesium, that are dissolved within the same plasma, because those are what was constituted plasma. So that's what constitutes plasma. Now, we migrated this to the cellular component. Cellular component, as you saw, we have red blood cells, which carry oxygen, thrombocytes, which will help prevent leaking of blood from damaged vessels. Then you have leukocytes, and we are going to discuss them further. Those that are granulotic, those that have granules in their cytoplasm, and those that are granular, those that do not have granules in their cytoplasm. Now, these leukocytes have various multiple functions, but what we know, what we can for now at this stage, we can just say they are involved in the defense mechanism of the body, white blood cells. The granulocytes are split into basophils, neutrophils, and eosinophils. What we are calling granulocytes. They are granular, you have lymphocytes and monocytes. So the lymphocytes are divided into the B cells, the B lymphocytes, and the T lymphocytes. Then the monocytes, you have the ones that are the ones that are what we are calling the mast cells that produce antibodies. So all these cells, the white blood cells, really their function is protection. Most of the granulocytes, they are the guys that arrive first. The monocytes, that's why you have macrophages coming from. So the cells that are involved in from the monocytes, you have the macrophages, you have the mast cells, you have the memory cells. So we are going to discuss some of them mentioned by white blood cells. So the leukocytes, the white blood cells, they are important, they are just important, their function is new. So you can see that even from this component of blood, you can see the functions. Red blood cells will transfer oxygen. Thrombocytes prevent damage of blood, prevent leaking of blood cells. Leukocytes are going to protect it. So that is the cellular component. Now, it's important for us to at least try to add a bit of this cellular component. We begin with the red blood cells themselves. Yes, Richard.
That 7% talked about the body, 7% of the weight. And I think also those substances which are suspended in the blood.
We are talking about the whole blood. So whole blood, two plasma and cellular components. So plasma cells protect the okay, if the blood is whole blood, that's the volume. What is the blood? What what blood is composed of plasma? Now this plasma is made up of what? It's made up of water, which is 99%. And from this water now this electrolytes that are there, you have all things that we specify like plasma. That's what the plasma. Then you have the cells on the other side. So now we come to the blood cells. Interestingly, the characteristics of red blood cells, they are composed of 75% water and 25% solids. And the primary solid in red blood cells is hemoglobin. This hemoglobin is the protein, the component by one that in a natural protein to make the this protein. So we have a hem and hemoglobin to make them together. They form this protein that makes solid body. Now, one interesting feature of red blood cells is serum and has no nucleus. Most red blood cells, they have no nucleus except for beta and some. This one is very important. When red blood cells are very much, when they are agreement of what is the production of blood cells in bone marrow, during the specialization and maturation, the erythroblast is used to or mixing erythroblast like blood cells. Before they what they get into the system, they need to yes. So our lives except for this. So we can get blood cells are all lives. So that's what it is. So if I get this sample of blood cells and scan you must say but if you get blood sample and that over and scan it, you must say the nucleus. And if there was excessive injury and it bled so much, and the bone marrow tries to break blood, that is when it ends. The red blood cells are so powerful. Just when you scan the but what you see is not a new. Just so immature those that are still laying that are getting then they might see that it was so much of a trauma. When an arm has a blade so much no bleeding, they will increase the sample. Double concave disc. Now, double concave disc, it gives it almost 2 or 3 red blood cells by concave. One is to increase the surface area for oxygen and carbon dioxide exchange, the issue of diffusion. 2 is to allow it to change shape. Red blood cells are able, because when you are biconcave, it's like you are twisting. The biconcave you are talking about is like you get the money rubber, and when you get that money rubber, you try to make an 8. That's how the red blood cells are. So that shape allows them to change shapes. Red blood cells, you are able to, they can be able to absorb water. Water can be able to diffuse, it will just become round, a whole lot of it. If it gets a certain maximum of water, but if it extends, it will be able to burst. So that is one of the features. That is why it is by one to allow it to change shape, to allow enough surface area, and also to allow it movement when it is moving, even the attachment of other constituencies. Now, we are saying these red blood cells, they are variable in sizes. I was talking about depending on the species, depending on the animal. And the red blood cells, you can imagine, interestingly, these guys, they've lost the nucleus. They also do not have the mitochondria, interestingly. But these red blood cells, in fact, even blood, the way it is, red blood cells, the way it is, they require energy, and they require glucose as their source of energy. So interesting is even when you collect blood in your vacutainer, those tubes that you use, that whole blood, as long as that whole blood does not clot, it is still whole. You find that actually the red blood cells within those, they will need glucose. So where do they get the glucose? Because at that point they've lost. They do not have mitochondria, they do not have a nucleus. So they use that's what the red blood cells use for their energy in order for them to survive. So those are the characteristics of red blood cells. But maybe we can go further. So the red blood cells, we are saying the component they have, one of the components they have is the hemoglobin. So it's composed of hem. So which is just a pigmentation, that is iron. And then you have globulin. So hemoglobin is just the iron plus globulin protein. Now, why is the iron there? Why is the iron there? The iron is there so that that's where the oxygen binds. If you still remember your chemistry, this is where now we start applying stories. Those stories we're discussing in chemistry, that's where they apply. So when we're talking about iron, that's where so the globulin is the one that is an attachment, it's a carrier. It will carry iron. And this iron, that's where the oxygen attaches. So every hem group can carry one molecule of oxygen. So in one hemoglobin, you have about four hemo groups that are attached to each globulin molecule. So one globulin molecule, one hemoglobin, it will carry about 4 oxygens. So the globulin is the one that is the protein that is produced by the ribose. So that is the component. So hemoglobin is just hem and globulin. Hem, that's where you have the component of iron. So that is the component of blood, the hemoglobin part. That is what it is made of. Now, hemoglobins is in 3 forms. What we are calling normal hemoglobins. You have embryonic hemoglobin, meaning an embryo. It has an that is what you find in embryo in the developing fetuses. Then you have also, when there's still just a mass, these cells that are dividing, they have their own, we call it embryonic hemoglobin. Then you have what we call the fetal hemoglobin. So it's found in fetal blood. That is in the last trimester, mid to last trimester, and even up to somewhere the months before, after birth, you find that you still have fetal hemoglobin. Then you have the adult hemoglobin that are found in red blood cells of all animals beginning a couple of weeks to a couple of months after birth. So those are the stages. So an embryo will have what your embryonic hemoglobin. Fetus will have hemoglobin, and when it is, you have what you are calling that's just a few months after is born. Now, function of hemoglobin, as we have mentioned, is just the issue of carrying oxygen. What we are calling oxyhemoglobin is a hemoglobin that is carrying oxygen. Then you have what we call the oxyhemoglobin. It is the one that has released. So we have those 2 functions. So it will carry oxygen. That is the most function. So oxyhemoglobin carries, and the deoxy is the one that has lost. It has no, it has lost, or it has transported and left the hemoglobin where it is. Now, the story of blood, and we are talking about hemoglobin, we cannot talk about oxygen. You are carried to the cells, so that within, when the cells are doing metabolism, you carry this oxygen to the cells, so that as the blood is coming back, it comes back with carbon dioxide. So the transport of oxygen, it is the hemoglobin that does the transportation. Now, when carbon dioxide is brought back, interesting is there are 2 forms. Indirectly, there is what we call an indirect method of how the carbon dioxide is transported, and the direct. So direct one is the one that we have focused on, which is where it just diffuses into the red blood cells. Directly into blood cells. And once it gets into red blood cells, it will be transformed into carbonic acid. This carbonic acid then is going to be ionized into hydrogen ion and bicarbonate ions. Now, the deoxyhemoglobin that we were talking about is the one that is responsible in the transportation of carbon dioxide. Transport it as carbon dioxide? No. It will transport it in ions. The deoxyhemoglobin will accept the hydrogen. Carbonic acid that has been formed, the carbonate, that's how it is to go into the blood. So once it is hydrogen carbonate in the blood, in the red blood cell, the hemoglobin that has lost oxygen, it only has access to attach, so it will get the hydrogen from the carbonic acid. And then the bicarbonate will diffuse back into the plasma. I hope the story is making sense. So carbon dioxide, when you are talking about, say, the exchange of oxygen and carbon dioxide into the capillary of the tissues or into the cells, what really comes back is not the carbon dioxide in the blood. What really comes back is the hydrogen that is gotten, and the bicarbonate is just diffuses into the plasma. So you find that you no longer have. So carbon dioxide is transported the hydrogen ion and is a bicarbonate ion from the tissue, and now it is in the red blood in the red blood cell. So the red blood cells are the ones that transport carbon dioxide from the tissue. But how do they transport it from the tissue? They don't transport it as carbon dioxide molecule. What happens? It will diffuse. When it diffuses, it will become a hydrogen is going to be added to make it a carbonic acid. Once it has become a carbonic acid, this carbonic acid, it will ionize. It will give you to hydrogen ions and a bicarbonate ion. So the hydrogen ion is going to